and welcome. Dear students, today we will discuss the topic fungi, degeneration of sex. Sexual reproduction is ubiquitous in nature. As the engine that drives genetic diversity, sex accelerates adaptation and removes deleterious mutations. As such, it plays a central role in the origin and success of species. The existence of some sort of sexual reproduction in fungi has long been suspected, but of late, observations have multiplied and facts accumulated, which leave no doubt of its existence. However, studies on the major groups within the fungal kingdom have provided significant and wide-ranging clues regarding the gradual loss in sexuality and sexual identity. Let's discuss the process of degeneration of sex in fungi. The lower fungi or Phycomycetes comprise the simplest and the most primitive group of fungi represented by about 1500 species. Sexual process ranges from isogamy to oogamy through anisogamy. Bringing together of male and female protoplasts or nuclei, that is plasmogamy, is achieved by planogametic copulation, gametangial contact and gametangial copulation. Now we will discuss them briefly one by one. Number first, planogametic copulation. This type of sexual reproduction involves the fusion of two naked gametes where one or both of them are motile. The most primitive fungi produce isogamous planogametes. Example, Syncytium, Olpidium, Plasmodiophora, etc. The anisogamous planogametes are only found in the genus Allomyces of order Blastocladiales. Monoblepharis illustrates the most advanced type of planogametic copulation. It produces small opisthokonte male gametes in large numbers inside the sex organ called antheridium. The oogonium produces a single large immobile female gamete, the egg or ovum. The male gamete enters the oogonium and fertilizes the egg. Number second is gametangial contact. Here, a group of aplanogametic lover fungi, that is Oomycetes, produce non-motile gametes that are never released. The male gametangium is called antheridium and the female oogonium. No sperms are organized. The gametangia do not actually fuse and they never lose their identity. The antheridium puts out a slender tubular outgrowth, the fertilization tube, at the point of contact with the oogonium. Meiosis is gametangial. Examples are Saprolegnia, Aquila, Pythium, Phytophthora, etc. Then we have the gametangial copulation. In another group of aplanogametic lover fungi, that is Mucor, Rhizopus, etc., gametic union is brought about by the fusion between the gametangia. The uniting gametangia lose their identity in the sexual act. The intervening walls between the two gametangia dissolve, forming a common fusion cell. Plasmogamy is immediately followed by karyogamy. The diploid contents of the zygospore or the diploid nuclei undergo zygotic meiosis. The meiospores on liberation germinate to produce a new haplomycelium. Thus, it is obvious that sexual reproduction arose several times in the evolutionary development of lover fungi. Sexual reproduction becomes less frequent than asexual and is resorted to towards the end of the growing season when conditions unfavorable for growth are to set in. The gradual transition from the aquatic forms to purely terrestrial species noticeable in the Phycomycetes culminates in Ascomycetes, which are fungi of drier terrestrial habitats completely lacking motile cells in their life cycle. It is seen that when we move from the primitive to the more advanced Zygomycetes Phycomycetes, there is a tendency for the sporangia to be transformed into sporangioles and then into conidia, the typical asexual spores of Ascomycetes. In fact, in some species of Ascomycetes, conidia alone appear to be sufficient for survival. Let us now turn our attention towards Ascomycetes and see how the degeneration and reduction in sexuality has progressed during the course of their evolution. The Ascomycetes are characterized by the presence of an ascus. The ascus is the product of sexual act and the ascospores produced inside it are thus sexual spores. The young ascus is always binucleate. These two nuclei undergo karyogamy to form the diploid zygote nucleus. This is followed by 3 successive nuclear divisions. The first division is normally reactional, the others equational, forming usually 8 ascospores by free cell formation. The various methods and mechanisms that bring the compatible nuclei together in the progressive process of reduction in sexuality vary widely in different groups of Ascomycetes. These include gametangial copulation, gametangial contact, autogamy, parthenogenesis, spermatization of trichogynes, and somatogamy. Finally, there is total absence of sexuality. Now, we will focus on these mechanisms and discuss each one of them briefly. We will start with gametangial copulation. This process may be regarded similar to that found in the zygomycotina. This persists among the primitive Ascomycetes, that is Hemiascomycetes. In this method, two morphologically similar gametangia copulate in the same manner as those of the Zygomycetes. Plasmogamy takes place as zygote is immediately formed at the fusion bridge and the zygote gets converted into an ascus. No dikaryotic phase is developed because plasmogamy is immediately followed by karyogamy. This is exemplified by members of the family Diplodescaceae. In the family Sacromycetaceae, however, modification of the above process can be seen as complete suppression of the specialized gametangia and copulation takes place between vegetative cells, that is somatogamy. The gradual suppression of the sexual phenomenon is characteristic of yeast fungi. The formation of ascospores in yeasts is contingent upon a sexual process. Of course, no sexual organs like antheridia and oogonia are produced. Instead, the sexual process is extremely simplified, however, occurs through plasmogamy, karyogamy, and meiosis. In Saccharomycodes ludwigi, the ascospores directly function as gametangia and copulate. Sexual fusion takes place within the ascus between the two adjacent ascospores of opposite mating types, that is plus and minus strains. In fission yeast, Schizosaccharomyces octosporus, each haploid somatic cell is a potential gametangium. At the time of sexual reproduction, two somatic cells come to lie side by side and each sends out a short, narrow beak-like process. The two meet and the intervening walls dissolve to form a conjugation tube, the latter functioning directly as ascus mother cell. Further modification of sexual process is seen in tephrinales, where any dikaryotic vegetative cell becomes an ascus. Moreover, the ascospores are capable of budding. The type of sexuality exemplified by tephrinales has several interesting phases, both of degenerate and evolutionary character, such as suppression of specialized gametangia, copulation between blastospores, development of dikaryophase, formation of ascogenous cells, delayed karyogamy, alternation of haplophase with dikaryophase, and production of many asci and ascospores within a single plasmogamous fusion. Next method is gametangial contact. This process, though similar to the oogamous type in its initial stages, has many evolutionary and distinct features. In this method, well developed uninucleate or multinucleate gametangia develop. The males are designated antheridia and the females ascogonia, which are provided with a specialized receptive hyphae, the trichogyne, not formed in the oomycetes. The male nucleus or nuclei that migrate into the female via the trichogyne do not fuse but remain associated, thus undergoing dikaryophase. Then one or more ascogenous hyphae arise from the ascogonium. and ultimately SI are formed by the crozier or hook method. This type of sexuality occurs commonly in taxa belonging to several groups such as Plectomycetes, Pyrenomycetes and Discomycetes. Dear students, let us illustrate sexuality in Aspergillus from Plectomycetes and see how it throws light on the degeneration of sex in Ascomycetes. Most species of Aspergillus are only known to reproduce asexually. The sexual or perfect stage is rather rare. Possibly, the species lacking it have lost it during the course of evolution. In some, there is a degeneration of male nuclei, whereas in others, the antheridium is rudimentary, non-functional or even absent. In Aspergillus herbariorum, the tip of the antheridium fuses with that of trichogyne, but there is no migration of contents of the antheridium towards the trichogyne. In other species like Aspergillus nidulans, the antheridium may develop late. By the time the ascogenous hyphae have already been developed from the ascogonium. In a few species, the male nuclei in the antheridium degenerate before the antheridium has reached maturity. The antheridium in some species such as Aspergillus flavus, Aspergillus fischeri, Aspergillus fumigatus does not develop at all. Only the ascogonia are seen to develop. In genus Eurotium, the antheridia and ascogonia are produced close to each other on the aerial somatic hyphae. The antheridium may or may not be functional, but pairing of the nuclei takes place in the ascogonium. When the antheridium is functional, its nuclei enter the ascogonium and pair with its nuclei. When the antheridium is non-functional, the ascogonial nuclei form pairs themselves. In Penicillium also, ascocarp formation is absent. Only a few species are known to reproduce sexually, for example, Penicillium vermiculatum and Penicillium stipitatum. Here, the mycelium contains uninucleate cells from which develops a uninucleate ascogonium. The ascogonium elongates and its nucleus divides several times to produce as many as 64 nuclei. Simultaneously, a uninucleate antheridium also develops and coils spirally around the multinucleate ascogonium. The antheridial nucleus does not migrate into the ascogonium. It remains very much there in the antheridium even after the development of ascogenous hyphae. This indicates that antheridia are formed but remain non-functional. In Erysippe of Pyrenomycetes, even the actual process of ascosporogenesis is not clear. In Sphaeriales, sexual reproduction takes place by spermatia uniting with the trichogyne of ascogonia. Many species bear antheridia but are generally non-functional. Class Discomycetes show progressive reduction in their sexuality. Both the antheridia and ascogonia are functional in Pyronyma. In Lechnia stercorea, the antheridia are non-functional, and in Lechnia critea, the antheridia are totally absent. Along with the total absence of antheridia, Humeria granulata shows the disappearance of trichogyne from the ascogonium. Some Discomycetes like Morchella show the total absence of both antheridia and ascogonia. The accessory parts of sexual process are completely suppressed. Consequently, the sexual process is extremely simplified. Plasmogamy in Morchella takes place by the following two methods. Number first, somatogamous copulation. Here, two vegetative hyphae of the subhymeneum region of the pileus come in contact. The intervening walls between the copulating cells dissolve at the point of contact. Later, the two multinucleate protoplasts intermingle in the fusion cell. The two functional nuclei, one from each copulating cell, then form a dikaryon. All other nuclei in the fusion cell disappear. Later, the ascogenous hyphae arise from the fusion cell containing the dikaryon. Each young ascogenous hypha receives a pair of nuclei. These are the derivatives of the parent dikaryon. The ascogenous hyphae afterwards become septate. The terminal cells of these hyphae function as ascus mother cells. Number second is autogamous pairing. In Morchella elata, any vegetative cell of the subhymeneum may establish a dikaryon. It is accomplished by pairing of two of its own vegetative nuclei, hence called autogamous pairing. The remaining nuclei in the cell degenerate. The cells with dikaryons develop ascogenous hyphae as in somatogamous copulation. The two nuclei in the ascus mother cell undergo karyogamy, forming young ascus without crozier formation. This is followed by meiosis and 8 ascospores are formed. The sexual apparatus is completely lacking in Pezziza vesiculosa. However, sexual reproduction is extremely simplified and consists in the association of two purely vegetative nuclei in a pair to form a dikaryon by somatogamous copulation of adjacent hyphae or by autogamous pairing. The third method is autogamy. Here, the female sex organ is retained and sexual development proceeds without any direct fertilization from an antheridium, as the antheridium is non-functional. The contact stimulus of the male gametangium is enough to initiate further development, and the dikaryon stage is attained by pairing between the ascogonial nuclei only. This type of development is seen in certain species of Eurotium like Eurotium amestylodam, Eurotium repens and Eurotium glaucus, Penicillium vermiculatum, Phylactinia corelia, Sphaerotheca morus uve, in many Sphaeriales and Pezziziales like Lecnia stercoria. Fourth method is the parthenogenesis. This is further modification of autogamy. In this case, a complete elimination of the male gametangium has taken place. Dikaryotization and the production of the ascogenous hyphae occur in one of the following ways. Number first, in Humeria granulata, the compatible nuclei of the opposite strain enter one of the nearby hypha by means of mycelial fusion. This enables the opposite strain to migrate into the ascogonium, which then gives rise to ascogenous hyphae in a normal way. Number second, in Ascobolus citrinus, nuclei from one of the adjoining cells enter the ascogonium by the dissolution of the partition wall. The ascogonium then gives rise to ascogenous hyphae and proceeds to develop asci. In Sartoria, which is the perfect stage of a number of aspergilli like Aspergillus fumigatus, the ascocarp is exclusively initiated by a coiled ascogonium and no antheridium is produced. Now, we will discuss another method, that is spermatization of trichogynes. This is highly specialized type of sexual process that is found in many higher Ascomycetes. Here, no antheridia are produced and the ascogonia provided with trichogyne are fertilized by means of spermatia, microconidia or conidia. This can be elaborated with the help of some examples. In Mycosphaerella, spermatia are formed internally in spermagonial cells. In perithecial Primoria, coiled ascogonia are formed, each with a basal ascogonial cell and a long trichogyne. After fertilization, the ascogonium throws off short ascogenous hyphae which develop asci by hook or crozier method. In Neurospora stophylla, the ascogonia are more degenerate and consist only of a coiled hyphae. which give out a number of hyphae, each acting as a trichogyne. No special spermatia are produced here, and the spermatial function is either performed by compatible conidia or microconidia. Sixth method is somatogamy. This is the final stage in the elimination of both the antheridium and ascogonium. The sexual process becomes extremely simplified. It takes place by the copulation between the cells of two compatible hyphae at any point along the mycelium through anastomosis. Two strains of the opposite sex, however, are necessary. Finally, there is disappearance of even this impulse towards sexuality. The two vegetative nuclei of adjacent cells of the same homothelic mycelium come together to establish a dikaryon. The two nuclei of the dikaryon fuse in the ascus mother cell to form a synkaryon. The later, by meiosis followed by mitosis, gives rise to 4 or 8 haploid ascospores within the ascus. Total apomixis, that is absence of sex, has been reported in some cases. In Ascobolus equinus, although solitary ascogonia are produced, karyogamy is totally absent. Only haploid nuclei are found everywhere. Only a single haploid phase occurs in the life cycle in which all nuclei are alike. This is indeed a rare and exceptional phenomenon. Thus, a gradual and progressive degeneration of sex organs is clearly visible as we proceed from the lower to the higher Ascomycetes. Dear students, now let us discuss sexuality in Basidiomycetes. In Basidiomycetes, plasmogamy takes place without karyogamy. The specially differentiated gametes and gametangia are usually absent in the Basidiomycetes, excepting rusts. But there are other ways of coming together of the two compatible nuclei. They include: number first, somatogamy or mycelial fusions; number second, spermatization or oidization; and number third, fusion between two oidea, which are small hyaline one-celled and uninucleate spores formed on oidophore tip. Thus, sexuality is said to be reduced in Basidiomycetes. The primary manifestations of sexuality, that is, the union of cells and nuclei, are obviously present in the Basidiomycetes, in spite of the absence of such secondary criteria as specialized gametes and gametangia. The compatibility mechanism, in fact, is much more highly developed in Basidiomycetes than the other fungi. Let us discuss the sexuality in Astylago and Agaricus to illustrate sexual mechanism in this group. No sex organs are developed in Astylago. The sexual process is represented by three fundamental phenomena characteristic of it, namely plasmogamy, karyogamy, and meiosis. In Agaricus and other mushrooms, the sexual apparatus in the form of sex organs is completely lacking. Their function has been taken over by the somatic hyphae, which are heterothelic. The fusion between two somatic hyphae of plus and minus strains represents the first stage. Plasmogamy is accomplished by somatogamy or somatogamous copulation. At last, we will take a look at imperfect fungi. Fungi imperfecti or Deuteromycetes comprise of a group of fungi in which only the asexual or imperfect stage is known. The sexual stage, also called the perfect stage, is completely absent. The sexual spores like sporangiospores, ascospores, and basidiospores are either non-existent or have not been observed or discovered so far. Reproduction takes place chiefly by the formation of exogenously developed asexual spores called conidia. Dear students, it is thus clear that there is a gradual reduction of sex as we proceed from lower fungi, that is Phycomycetes, through Ascomycetes to Basidiomycetes, and finally in Deuteromycetes, sexual stage is completely lacking.